So thank you again for having us. It's important for Revvity to be here. Maybe before starting, actually, before today, had anyone ever heard of Revvity? Who did not know about us? Let's start with this. Okay. I'm actually surprised. I was expecting more hands rising. Okay, so good. Revvity, for those of you who do not know us, bear in mind that it's okay. It's a 2.5 years old company. Revvity is a contraction of rev, revolutionizing, and the Anglicism of VT for life. As much as a company is young, it's actually a large umbrella that covers many companies that you may know working in life science and global health. So this company actually covered from fundamental research all the way to diagnostics. It carries many companies that provide with reagents, instruments, softwares. And if you don't have them in-house, we have our own R&D using with those 3 pillars, we can provide service and we can just hand out the results to you. So we partner with internally and also externally. We are 11,000 employees globally present on this planet with one goal, which is improving life. So improving life, what it is, it goes from partnering with clinical labs, but also fundamental research, academics, governmental organization, and pharmas and CRO. That is the truth. So what is this company doing? It's 3 main pillars, and for the sake of this presentation, I'm gonna actually make a gross summary. We do multi-omics analysis, we do biomarker identification and prediction, and for what I'm gonna be talking about today, cell imaging. If we need to go just quickly on the diagnostic part, because I will not extend onto this, Revvity is 40 million babies every year tested when they are born for health-threatening condition. It's 110 countries. Egypt is one of them. It's 20 years of experience in testing TB. And during COVID time, in between our reagents, automation instrument pipeline, we tested 75 million people. So we tend to do things, and also in live imaging, in cell imaging, and in research environment, sort of at high scale. So stay with me. The formal presentation is this one. This is my slide with my email address. If you need to send me an email, there is my LinkedIn profile. I post rarely on social media, but the little thing I post are usually microscopy related. And the funny part of it is what I actually do for a living. So actually my life is not very different of when I was a scientist. I spend most of my days having coffee, discussing science, experimental design. This is what I do most of my day. Then I play on microscopes and I take pictures of funny samples. And I realized only yesterday morning the irony of the food chain here. Anyway, I barely ever publish paper any longer. There was a talk this morning. I'm not even in the acknowledgement. But I do now prepare technical notes. It means that those PDFs are available online. When you don't know how to implement an experiment, look, there might be some protocols ready to use for you to give you ideas on how to implement them. So, what do we do at Revvity in terms of imaging system? I'm going to talk to you about 3 main instruments. We do automated microscopes. We do systems that can go from the cell level. So this is a cell image cytometer, meaning that I image every cell in your well. So you have statistical power out of it. But if your experiment requires more subcellular information, subtle morphological differences to be pictured, then we may have a subset of cell image, but we will also need more flexibility in terms of microscopes, so that the 2 on the right-hand side. So, Revvity is thinking in terms of workflow. As I said, we tend to go from a reagent all the way to instrument and software. We think every question at the global level. And to give you an idea of how we prepare things, we work at those 4 colors level. For today, we have 10 minutes. I'm going to focus on the yellow, focus on the purple. I will not talk about all our analysis softwares, and I will not talk about automation capabilities. But what can we say? First of all, half of my slide is covered by sample preparation, and this has been highlighted already enough this morning. It's not 10 minutes, it's not a coffee break, it's a full day if we go into the steps. But I'm going to take this with 4 take-home messages. First of all, when you have a protocol, that might be funny, but a protocol has to be followed per steps. And this is maybe important, obvious for a few of you, but you don't understand how many times I have to explain you missed a step. If you don't fix, if you don't permeabilize, your antibodies are not going to get in. That happens every day. Second, when you work in microscopy, you actually need to count the cells you're about to put into your assay. So cell counter are very important. Third point, when you are seeding cells, you're most likely seeding them in tissue culture plate onto cover slips. The fact is all our microscopes are automated. I no longer break cover slips. I actually work in those plates, which we call macro imaging plates. They are plastic-based, but the bottom is as thin as a cover slip, meaning that you seed your cells in there and they stay there. You stand, you fix, and you image directly from this plate, meaning that if you have a very low count of cells, it makes every single cell count. And then when it comes to dyes, how are you labeling your cells? Well, if those are alive cells, most likely you're going to need vital dyes. If you have fixed cells, you can go with organelles dyes. If you have a protein of interest, you're going to go with antibodies. We have ready-to-use kits, which comes with the protocol that you have to follow. And most of our kits actually ship desiccated, meaning room temperature, which I think is important. Second part of this presentation, and the one that matters for us today, imaging and analysis. And you might know this expression, an image is worth 1,000 words. I like to make it slightly more scientific. An image is worth 1,000 descriptors. And we've been talking about microscopy and what an image is. In my word, I don't even think it's worth to go to a microscope if I cannot analyze the image afterwards. So it means that in our case, all our instruments actually are piloted by a computer. The computer has an analysis software. The 3 instruments I'm going to be talking about today all have the analysis software attached. So actually what you get out of the microscope are not so images, it's your Excel sheet. So I made an effort of trying to put them at scale, and this is me. For those online, I'm 1.60 meters, just to give you a reference. And so this is how we work. So let's start with something simple. You come to me and you tell me, right, I have cells. Well, cells are already very informative. We could be starting with counting the cells, because depending on the treatment you're applying, actually, the number of cells could vary. The Celigo is what we call an image cytometer. It has a theta lens. It images absolutely the entire well, whatever the format of plate you feed in, meaning that every absolutely single cell is detected, is counted, and segmented. Now you're telling me, yes, I want to see my cells alive. So in this case, you're going to need an incubator. I will need to change gear and put your cells into my 2 other microscopes that have inside an incubation chamber. What can you do on those? Well, first on left-hand side, it was supposed to move. It's not moving anymore. They were? Okay, let's replay the movie. So this is a kinetic, overnight kinetic, an image every 10 minutes. What you see are the traces of the image analysis software. I could show you the images, but what is important here is actually the analysis. On the right-hand side are transmitted light, and it's cardiomyocyte. So this is what we call fast kinetic. In this case, I can even look at the cells moving, and this is sufficient for me to trace the beating of those cardiomyocytes. We have also integrated what we call commercially phenologic AI. We can do nucleus and cytoplasm segmentation label-free with our AI model nowadays. Let's move to another gear. I know that the continent study heavily host pathogen interaction. When it comes to this, where can we start? Well, we could start with just live dead dyes, and you can count the cells as surviving or dying upon infection. We could go a bit further. If your pathogen has a reporter protein somewhere, we could actually track the cells that are infected. That could be part of what you're checking. If the host variability, the pathogen variability, or whatever treatment you are implementing, just by looking at the reporter protein and counting the cells that are positively infected is going to give you an answer. If you're telling me that your pathogen has a slightly more complex life cycle, doesn't completely kill the cells, it's more complicated, okay, we're going to have to change gears. In this case, I'm going to have and need high magnification lenses, high numerical aperture lenses, and we can go further. So the first one in the center is the trypanosome. So you're going to have cells infected. I can count every single bug and count total number of bugs per cell. I do that with trypanosome. I do that with plasmodium. I do that with mycobacterium very regularly. Those of you who work on pathogens that can live freely, you have here an example of E. coli. Those cells grow directly. What do? You treat them and you see whether or not they survive. This assay is what we call a cell painting assay on bacteria. And in this case, what they do is antimicrobial resistance screening, trying to find new compounds to get rid of those little bugs. We quickly alluded to it as well. That will be the third example, 3D, because we are beautiful 3D samples, right? There are two means of growing 3D samples, I would say, just to make it short. We have first the concept of growing them in flat bottom plate. So it's going to be a flat bottom. You put an hydrogels. You see your organoids growing more or less everywhere. With a Celigo, I can look for all of them. For biobanking, you have a lot of people doing this type of assay. We can obviously look at them also in U-bottom plate. In this case, you grow them in a little salad bowl, and it's like a plastic with low adhesion. So the cells aggregate. It forms one ball per well, meaning that we can look at escape artists if you do some invasion assay, or you can label them with dyes, viability, apoptosis, hypoxia, name it. This is actually a way to read out very quickly what's happening into your cells. If you need to go deeper into the 3D samples, this is solely when I will gear up into confocality, solely at that stage. So in this case, what do we have? The first layer of images show you in the flat bottom plate, you have your hydrogels, your little organoids floating a bit everywhere. You need to find them for 3D imaging. So we have a procedure that allows us to find them in X, Y, and Z, and to reacquire only the sample with Z stacking. And once you have generated your Z stack, you can redo your 3D reconstruction. So this is now the lower panel. With a 3D reconstruction, I can do single cell segmentation of the little guys on good and prepared, well-prepared samples. And if you have labeled, for example, with Ki67, proliferative index, I can do a proliferative index into a 3D sample. It was mentioned today that we're talking a lot about human samples. So for those who did not hear about their topic of research, I tried to add an example of marine biology, vegetal biology, people studying bugs, like this is a fly. And there are some cats for those doing some mouse work or tissue work. With this, I should be more or less on time. I thank you for your time, and I think it's a session for question now. Thank you.
Thank you very much, Laurianne. All right, so I think we can quickly deal with a couple of questions for our speakers, and then we're going to jump straight into the community discussion. Any immediate questions for Laurianne, Christina, and Karen up front?
Yeah, I want to ask Laurianne about her last slide. You mentioned there the U-bottoms, the U-bottom dishes can be used in confocal, etc. Is the curvature of the bottom not interfering with the light path? Because I would have thought, you know, the perpendicular level of a flat bottom would be necessary.
So in our case, the curvature of the bottom of the well has an impact on the autofocus routine, but it's something we can actually circumvent. We have a routine to pick that up.
Okay, thanks.
This is for Karen. I have just a general question about these open scopes. How do you validate them? How do you check if they are good enough or they compare to commercial ones? Uh,
I love that question. Thank you. It depends on how much money you have. If you are like we are, sort of skilling, kitting ourselves up as a bit of a hub to build and send out systems, there are calibration sites that you can buy, various masks that you can get to make sure that they're appropriately aligned. We're very aware that that costs a lot more money than most labs would want to or be able to put into this. So one of the things with our low-cost systems that we're doing is actually twofold. One is developing low-cost, for example, 3D printed masks that can be aligned and put on system-specific, slot this in where your objective comes out, make sure that light is coming out central on them, and that's a way to get it aligned. In terms of QC of the image quality, interestingly enough, the global community is increasingly aware that, let me think of the best way to frame this. You can get very high quality calibration slides and ways to do this, but using regular standardized slides is a good way to do this. So for example, the fluo cell slides that Invitrogen sells, there's a group in Canada working to benchmark resolution and image quality of commercial systems with those slides, and the exact same approach can be taken with these sorts of systems. It is critical to do it, which is why I love the question, because even with your commercial systems, if a vendor tells you, oh, the system can give you 50 nanometer resolution, don't believe them, test it. And the best way to do that is with a sample and checking, and you can take the exact same processes with the system. Yeah.
For the IVF microscopes, is it any difference for clinical and research types? And do you offer some technical training for it? Because you have one, and we lack some training for it, and also the compatible reagents for the microscope also. So do you offer such?
We're going to have to discuss together then, because yeah, usually, so instrument, we provide training at installation, and otherwise we can do repeat training. So it's maybe something we can discuss in your specific case.
I have a question for both of you. So maybe I start with Laurianne. The operetta. I mean, now it's better. Okay, so cool. Who is the distributor and also the maintenance person for operetta in North Africa? And I know the distributor in Cairo. I talked to them, but it's very different. Are they here?
Yeah, he is outside. So you discuss with HTDS today, right?
No, not yet, actually. I want to discuss more in detail. I'm very interested in this microscope, but in general, we are having problem with this microscope in Europe in terms of maintenance. I want to know what is the plan in Africa, like who maintains this microscope?
So those type of instruments are extremely robust. So we actually plan usually one service maintenance per year. So then it's the service engineer traveling.
Yeah.
So in this case, we would just need to know who is trained in your local area, and yeah, just identify the person who can be traveling to your place for maintaining such a system.
Yeah.
It's once per year.
Maybe I can talk to you off the conference specifically. So now I love the open system. I just want to know, when you say you train scientists to use it? Do you also train them to build and play around with the units enough so that it's an independent kind of Lego system where they can work with it? And do you think also in the concept of ABIC in general, the idea of integrating 3D printers to solve problems, to get spare parts that take months to come to Africa, do you have such a facility in several nodes in Africa that can help reproduce the commercial parts into 3D printed units that are also modular?
Thanks for the questions. Currently, no. Planned for, yes. So you just identified some key components of what we're building into what we're hoping to get in terms of the next round of funding. So we're in the training that we have done with users so far in Cape Town, and Mike, feel free to jump in about this as well, because you were actually more heavily involved in the earlier stages than I was, but it was very much an exposure, and this is how you could build your own. But a key aspect of this is if you've configured it, if you've built it, you know how to maintain it and replace components because it's modular. One of the hardest things, especially with the system so far to do, or this ecosystem, has been to work out, if you're completely naive, which components you need, how to configure your own system. So the group at Imperial thus far has been heavily involved in the configurations of most systems that have gone out. With our packaging of the mobile system, the hope is, because it's kind of something pre-existing before it gets shipped, we've solved that problem. And the how to configure it and expand it to make it something that you need in your region is exactly what we're building into the next phase of the project, fully equipped with within a region, a 3D printer, and funds to, if you need something properly machined, to do something like that as well. Again, goes back to, this is a lower cost way to do it, and we're hoping to demonstrate that this is a sustainable way to do imaging and this sort of work. It's not free. All of this does cost money. Yeah.
Can I add a thought, in the sense that there's certainly economies of scale. So you might be able to build and machine your own open frame components and build your own microscope if you have the capabilities of doing that. But how do you share that capabilities and make it scalable? There is nothing stopping us as a community from, and as individual community members willing to take an opportunity of starting a company that can help produce these components potentially. And this has always been the ambition of OpenScopes. So OpenScopes has a name, but it's not a brand. It's not a company, it's not a product. There is nothing stopping somebody who knows what to do and knows how to do it and has identified a market from establishing a process that helps manufacture what is needed for OpenScopes locally so that it's cheaper. And then you've got an opportunity to capitalize on that economy of scale. And you've got the expertise so that it becomes a little bit more standardized and centralized. There's nothing stopping that from happening. It's something that we've always dreamed could potentially happen, but we need to find how to do that and who could do that. But that's where ABIC is also meant to play its part. And where is the opportunity for this kind of thing? Does it exist? Is it feasible? Is it not feasible?
Yeah, so I'll check on that, I guess. Critical things for the projects they are is improving the accessibility of documentation and training. And a big thing about the project that we're trying to do is actually, for the first time, at least in a sub-Saharan setting that we've seen, is very actively pairing the biologists with engineers and developers. Because I was amazed by the talk this morning about this this faculty and department, that it is a pairing of engineering and science. We tend to not talk to each other like that back home. So we're actively trying to address that.
Hello, Adelaide, are you there? Are you ready for a question?
Yeah, thank you very much, Mike. I believe I can be heard by everybody.
Yes.
Yes.
All right, thank you very much. Well, what I had in mind was to be directed to Caron, and but I think the last contribution from Mike is trying to make it clearer to me. I was worried if the issue of open scope microscope, which looks very interesting as presented by Caron, ridiculously cheap, so to say. But I was thinking that low-income regions where people may have interest to actually procure those equipments. One big challenge that can come up can be shipments cost. But I think the clarification that Mike just gave now made me know that it's actually not a company, or it's what regions can come up with themselves. But however, if there are still maybe hardwares or components that we have people that produce them that can be taken to different regions, I was of the opinion that if those set of people can have a hub or like a warehouse in areas like Africa, that will make it closer to our people. That was my concern. Did you get my message?
Sure. Thanks, Laurianne. Yeah, so I will reiterate kind of what Mike said. So this technology is open source. Most of the frugal microscopy approaches and frugal science in general approaches are. We're working on demonstrating independent manufacture of key components within the next year. So that things don't have to be imported from the UK and Europe. The electronics and some of the optics are harder to work around. I'm not really aware of any optical manufacturing for microscopy on the continent. So that's the sort of thing that you would have to import from Europe or China. A lot of the electronics these days come from China. But there's nothing stopping individuals or groups of individuals wanting to set up something like this. And I think it's something for opportunity, but it's also something that requires resources and either startup capital as a business or investment from funders for something like this. And that's something that we don't yet have.
To add to that, thank you, Laurianne. It's a very different concept of having to replace a commercial microscope component, whether it be a Revvity microscope, a Zeiss microscope, a Nikon, whatever it might be, versus just the simple individual component. Amazon, Alibaba, they've already figured out how to do this shipment stuff at mass scale. So we should just leverage that. There's nothing saying that we have to be reliant on the commercial supplies for these kinds of things. The commercial supplies offer a wealth of other things that we mustn't lose sight of. But if the situation is appropriate and it's suitable for us to build our own microscopes, maybe we should look at doing that.
I will agree, especially cognizant of our commercial partner in the room, who is also a very old friend of mine. So I know, but there are times and places where a frugal approach makes a lot of sense. Fundamental technology, it makes a lot of sense. That gives you the opportunity to test, get proof of concept data, to write the grant, to get the more advanced system that would take you years to build. And that's, I think, where the more advanced approaches, more sophisticated approaches, and all of the support that comes from having an application scientist, tested, verified protocols, all of that stuff that comes from a commercial partner is incredibly valuable. I don't think we need to be at odds. I think the content is huge. There's so much room for all of us. But yeah, it requires, one way or another, requires resources.
I was going to say something, but now I've forgotten. I'll pose a question to the audience to kind of loop in the earlier session as well. And I'm going to ask a question for everyone to think about. What do you think the benefits or disadvantages would be if you managed to establish a core facility that was exclusively offering open source microscopes? In general, in whatever your setting may be. So Laurianne, Ame, if you're still online, if you had a core facility like establishment like what we saw Lisa and Ame talk about, where you were offering fee for service, what do you think the pros and benefits would be if you had 5 microscopes that you had built yourself? Do you think it would have been cheaper? Do you think it would be easier, more difficult?
Hi, Mike.
If you, yes.
Can I give a point?
1 second, I want to repeat the question.
Yep.
If could establish a core facility that offered microscopes, but only open scopes or something that you've built yourself and not an instrument that was from a commercial supplier? That's the question. All right, over to you, Ame.
Okay. I think during my presentation, one of the key challenge I highlighted was the lack of technical people for maintenance. As you know, with the open scope microscopes, you know all of the different parts of the microscope, so you can easily fix what's going wrong on such instruments with no need to bring someone from outside your country. When Liz was presenting, we noticed that one of her key expense was about maintenance. And during my presentation, I also highlight the fact that with SPS from South Africa, the bill they brought us was over 40,000 US dollar for a 1-year maintenance. So in our context, price of maintenance, people for maintenance is one of our key challenges. So having an infrastructure with open scope microscope could resolve partially or maybe totally these aspects. Over to you.
Thank you, Ame. I don't know how to respond.
Can I say something, Mike?
Okay, we'll go with Adeladoon and then we'll have 2 responses from in here and then we need to call the end of the session. So over to you, Adeladoon.
All right, thank you so much. It's a very interesting question to me. And just like Hamea said, for me, it's going to be serving 2 purposes if that kind of opportunity comes up my way. The first is that, number one, your question is, will it be cheaper? Yes, it's going to be cheaper. You have that thing at your doorsteps. You are the one doing it. And so it's going to be cheaper. The second advantage that I think that will bring is empowerment and development. You know, people will be involved in that kind of science, in the know-how of how the microscopes are being built. So 2 purposes will be achieved: the low cost and also empowering people, people learning in the job and getting to know how some of all these things work. So I think I also support what Amir said.
Thank you, Adeladoon. Lisa?
My answer would be layered with quite a lot of ifs. If this, if that, if. Because our biggest challenge is to have the people and the time available for support. If the microscope is easy enough to use that the majority of your users can be trained very quickly and they use it safely without breaking it, by all means, definitely. I agree with the 2 of the speakers already about the maintenance. If the training includes how to properly service that equipment solely by yourself, that's another if. Yes, absolutely. And then the other thing is breaking, because it builds so easily, does it damage so easily? Because all of our microscopes that we have are all fairly boxed. I think the only thing that you really can damage is the objective, and I'm very strict about objectives. But now there's lenses and lasers and alignments and stuff, and can it break fairly easily if you have all your users using it or misalign or something? So that's another if. Yeah.
All right. I apologize. I'm going to end the discussion there because I want this to continue afterwards.
Fine, I think I need to sit.
Over to Maya.
So I'm gonna just build on what Lisa is saying, and I'm just gonna add is what are you going to use the microscope for? So don't tell me you're gonna build a UC2 and use it for quantitative research and quantitative imaging. So choose the microscope that you want to build, know the application. If you want a research microscope, it should be robust to do a quantitative research. So this is very important, just the application, and then definitely it's cheaper.
So I think
Augusta.
it's clear that you can argue until the sunset on either side of this question. And I think it's up to us to start having these discussions, right? So hopefully we can continue this in the coffee break and potentially into the dinner.